So, hi everyone. Today's our topic is the Meselson Stahl Experiment, which was done in 1958. This experiment have been done that establish the fact as a truth in the field of the life science as well as the molecular genetics. Then, what Meselson and Stahl did as an experiment to prove that the nature of the DNA replications. Whenever the transfer is being done from the one generation to the next generation, they definitely involve the DNA duplications as well as the DNA replications. They prove the ideal of the DNA replications and what type of the replications is actually true. So there are three types of the model in the DNA replications. One is conservative replications, another is semi-conservative replications, and another is dispersive replications. In one process says that where the parental DNA remain as it is the same of the new DNA, like this. This is called the conservative replications. Another is the semi-conservative replications, where that the true strand of the DNA, where the two strand of the DNA, sorry, where the true strand of the DNA separated and each of those strand then served as a template for the synthesis of the new DNA strand. Dispersive replications. Another theory states that the dispersive replications, where the parent of the double helix is broken into the double-stranded DNA segments, and the segments then resembles into the complete DNA double helix, each with the parental and all the newly synthesized dsDNA segments interspersed. So, the Meselson and Stahl prove in 1958 which process is true actually. To understand the process, they conducted the experiment with the help of the E. coli. So we all know that the E. coli is is generated very first and as well as their generation time is only the 20 minutes. So which was the step of experiments. The first primary thing that they need to think before doing this experiment is to distinguish and differentiate between the parental strand and the new strand, so they can easily tag the bases. They did the bases with the tag and sorry they tagged the bases with the heavy nitrogen. So we know that N14 is a natural nitrogen or the light nitrogen, and N15 is a heavy nitrogen. They took on set of E. coli and cultured it in the heavy nitrogen medium for many generations to make sure that the E. coli receive all the heavy nitrogen. So they then they transfer the E. coli into the light nitrogen medium, allowed to divide them. So they want to prove the three types of the replications which was the true. Then they figure out is to find out their genetic material and to run in the jail to find out their exact pattern for looking at their DNA of the genetic sequence. Then they need to find another way to equal in the E. coli that the DNA to carry the whether heavy nitrogen or the light nitrogen that process that is present in the E. coli. So they did a very simple process in the cesium chloride gradient centrifugation. It is densely gradient centrifugation. It means the heavy density at the bottom and the light density or the low density at the top. So they loaded those DNA into the gel. The DNA of the E. coli, sorry, with heavy nitrogen ran it on the gel. They found only one band appear at the bottom, which heavy region due to the densely gradient, like that. Here is a one brand appear in the bottom. So after that transferring into the light nitrogen medium. Then they loaded on the gel the DNA of the E. coli in light nitrogen medium. They found two separated band. One is a heavy band and one is a medium band, like that. And after 2 or 3 generations, they look the DNA from the in light nitrogen medium, and they took the DNA from the light nitrogen medium and they ran it on the gel. After that, they found the three band: a heavy, a medium, and a light band found. So they resulted that if the model is a dispersive, then all of the band of the dispersive model should give you different density, but there you never find the heavy or the medium pattern. So this model is not the true. If it become conservative replications, we found one heavy and one light distinctively. You should never found a medium band. So it is obviously the semi-conservative replication, because we found a heavy band for the heavy nitrogen medium and one medium band just in the semi-conservative DNA replications. One heavy strand and one light strand present in the new DNA, and then they together give the medium band. So that's why we said the Meselson Stahl experiment prove that the semi-conservative model is the actual way to say that the DNA replications, and where the one parental strand acting as a template strand to produce the new DNA strand. Thank you.